Hello friends, welcome to some biology class. Today we shall discuss about competitive inhibition of enzyme. In this type of enzyme inhibition, the inhibitor closely resembles the substrate. That means the inhibitor and the substrate are structurally related. Because of this, the inhibitor compete with the substrate for binding at the active site of the enzyme. Because of this, such type of inhibitors are called as competitive inhibitors. These inhibitors are regarded as substrate analog because they closely resemble the substrate. The competitive inhibition is also called as substrate analog inhibition. The degree of inhibition is proportional to the relative concentration of inhibitor and the substrate. This type of inhibition can be overcome by increasing the concentration of substrate while maintaining the concentration of inhibitor at a constant level. In case of competitive inhibition, the maximum reaction velocity remains unchanged, whereas the value of Km increases. What is this Km? Km is the Michaelis-Menten constant. It is equal to the substrate concentration at which the reaction rate is half maximal. In case of competitive inhibition, the enzyme either bind with the substrate to form enzyme-substrate complex, or it can bind with the inhibitor to form the enzyme-inhibitor complex. But the enzyme do not bind with the inhibitor and the substrate simultaneously. This figure explains the process of competitive inhibition. This is the enzyme and this specific site is the active site. Because the inhibitor closely resembles the substrate, so they compete for binding at the active site. The binding of enzyme with the substrate leads to the formation of enzyme-substrate complex, but when the enzyme binds with the inhibitor, in that case, enzyme-inhibitor complex is formed. But in case of competitive inhibition, the formation of enzyme substrate inhibitor complex never takes place because the inhibitor and the substrate compete for binding at the same site. Let us take some example. Succinate dehydrogenase is an enzyme of the Krebs cycle. This enzyme supplies electron to the electron transport system. Succinate dehydrogenase catalyzes the oxidation of succinate to fumarate. There are a number of organic acids like malonic acid, glutaric acid and oxalic acids which are structurally related with succinate. Therefore, these organic acids compete with succinate for binding at the active site of the enzyme succinate dehydrogenase. As a result, this enzyme is inhibited. Out of this organic acid, malonic acid closely resembles with the succinate. The difference between succinate and malonate is only a methylene group. Because of the close resemblance between succinate and malonate, it is the most efficient inhibitor of the enzyme succinate dehydrogenase complex. Another example is that competitive inhibition can be used for the treatment of persons, those who have ingested methanol. Alcohol dehydrogenase is an enzyme that catalyzes the conversion of methanol into formaldehyde. And formaldehyde is highly toxic. It damages various tissue systems in our body, particularly optic tissue, that result in blindness. In case of methanol poisoning, through an intravenous infusion of methanol, the severity of the poisoning can be reduced because ethanol closely resembles methanol. Therefore, ethanol will compete with methanol for binding at the active site of the enzyme alcohol dehydrogenase. As a result, the rate of formation of formaldehyde will decrease and the methanol can be easily extracted by the urine. Friends, this is about competitive inhibition. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much.